Hey, science research. Welcome to another week of online learning. This week's plan is kind of short, um, just as it was last week. So for Tuesday, uh, Tuesday, May 5th, there are two short Edpuzzle videos. One of them is the video you're watching right now, JMol Structures Recap, and the other one is a little background as to why we learn JMol and what the importance of JMol is. And then on Friday, you'll have another Edpuzzle video, a real short one on chromosome 22. These are all short videos, by the way. And then there's a little JMO modeling activity and a brief molecule of the month article on myoglobin that you'll take a look at. And I have just a few questions that you can answer that you'll find right here on the spreadsheet on the Schoology sheet. Again, Edpuzzle link is right here. Click on this link and you just jump right into the class and you'll see the videos in there. Moving forward, we're going to kind of wrap up protein structure this week. And then next week, we're going to get into, just to show you guys a little bit of the plan. Next week, we're going to talk a little bit more about protein crystallography and what synchrotron technology is. And then we're going to start looking at the coronavirus. There's a lot of protein work going on with coronavirus. So over the next couple of weeks, you can kind of see, you can look ahead. We're going to take a look at it in a little bit greater detail. So this way you are well informed about it and you can pass that information on as well. And again, lots of short little activities that are, pretty powerful. So let's just talk real quick about the structures that I had you guys look at. And so this is a quick recap. These are the functions of each protein. So if we go back to the first week that we went live, one of the proteins you looked at was this protein called 1AL1. It's called alpha-1 synthetic peptide. Synthetic, it's a man-made protein. They went in the lab and they put together a bunch of amino acids, not just randomly, they did some research on it. And this is the structure that they had. And so you can see on the left-hand side, this is the structure that you would have produced. The use of these peptides is mainly for the inhibition of enzymes in a variety of diseases. So what they've been able to do is take protein sequences, put them together, and create novel small molecules. Okay, novel means new, and these small molecules can be utilized for the inhibition of different types of proteins. So one of the things that they're even looking at with COVID-19 is trying to develop some sort of molecules that can act as inhibitors for different types of enzymes within the virus and also on the outside of the virus, so they can neutralize it. Okay, what's interesting about this alpha-1 synthetic peptide is that it forms hexamers. And so on the right-hand side here, there are 6 of these. 1, 2, 3, 4, 5, 6. There are 6 of these hexamers. Here's that little sulfate ion, I'm sorry, which is right over here. And so they're all interacting this way, and there are 6 of them here, okay? And this is a neat little protein that, again, was synthesized. If you go to the PDB and type in synthetic peptides, there are all kinds out there. So scientists are really doing a lot of work trying to develop novel small molecules that can be utilized potentially as treatments. Okay, lysozyme. This is a real special enzyme. Lysozyme is probably one of the most widely studied enzymes out there. It was the first enzyme to ever have an X-ray crystal determined from it. So what they did, so this next one is lysozyme. And lysozyme is a fascinating protein. It's a unique protein, and it's very special in many ways. It's one of the most widely studied proteins out there. It's found in our tears, saliva, sweat, and other body fluids. It has a great ability to destroy a wide variety of bacteria that can enter our body through these various passageways. This protects our eyes from eye infections. It's a nonspecific defense mechanism, broad spectrum, that's very powerful. Lysozyme was the first ever X-ray crystal of an enzyme. So in 1965, David Phillips determined what the structure of this enzyme looked like, and he revealed it at a lecture at the Royal Institution. And you can see a picture from here. He's got the actual structure here that he made himself out of metal. And then what he also did was from the ceiling of this lecture hall, he hung the polypeptide sequence all the way down so that they could see what this looks like. But on the left-hand side, this is the image with the electron densities shown here. So we can see where the different aspects of the electrons are for all the side chains. And again, here's that yellow hydrophobic region here that's at the bottom of the cleft that forms the active site. So this is the active site right here, okay? And again, real fascinating enzyme. Lots of work's been done on it. We are going to work on lysozyme. If you choose to continue on in research too next year, we're going to spend some time crystallizing lysozyme and making up some trays. I want to show you guys how to create protein crystals. Real fun stuff to do. And again, this is what we'll be doing at Brookhaven Lab, working with crystals. And the first one we always start off with is lysozyme, okay? All right, so moving on to the next one, the zinc finger. Zinc fingers are real interesting. They have the ability to bind to the DNA, and what they do is they fit right into what's called the major groove of the DNA. What they do is they grab a zinc ion and they fold tightly around it. So here's the zinc, right, and here's that protein folded tightly around it. And when you did this, you see it kind of zigzags. It goes down, up, and down, okay? What a zinc finger does is it actually looks like a finger going into a groove. And what it does is they're short. They're like 20 or 30 amino acids each. And this one happens to work as a trimer. So there were three of them. There's one, two, and the third one's kind of tucked behind here. They're very common in plants and animals. And basically their job is to function in DNA recognition. They were originally discovered in the transcription factor, TF3A, which comes from frog eggs. And what they know is that zinc fingers are involved in regulating the gene transcription that occurs for the production of, in particular, for the frogs, it was for ribosomal RNA. But a lot of our genes are regulated by proteins like this, and they tuck very nicely into this groove of the DNA molecule. Okay, and so what I did is I took the same structure you had, and I left all three of them there, and it just wraps right around the DNA, forms a nice little structure. Next up is OpiA. This is the outer integral membrane adhesion. This is interesting. This comes from the bacteria Neisseria meningitidis, which produces or causes meningitis. This is a fascinating protein because what it does is it actually is embedded in the membrane of the bacteria. So here's the bacteria, right, Neisseria meningitidis, and what it does is it has this outer region here, okay? And this is an outer region that docks with different types of proteins on the cell surface of humans. So this actually functions as almost a key that allows the bacteria to get itself into the cells. Once it gets into these cells, it starts to reproduce rapidly and it wreaks havoc on the body. And what's interesting is this gray area here that we have, this is all hydrophobic, okay? It repels water. And so what that does is that sits in the plasma membrane. So just imagine now that, this is the outer layer of the cell membrane. This is the inner layer of the cell membrane, okay? It's a lipid bilayer. So this portion of the protein sits in a nonpolar region, and these chains out here stick outward, and they act as a docking mechanism for the bacteria to get into the endothelial cells, right, which is really fascinating. And that's how it infects the nervous system. And meningitis is a major inflammation of the meninges, which is the layer of membrane that surrounds the brain and the spinal cord. Can be very, very dangerous. Bacterial meningitis is extremely dangerous and can often be deadly within 24 to 36 hours. Okay, and then what we have highlighted here are all the different side chains that I had a group of students years ago look at this, and they identified all the important side chains that were involved in docking it. And if you look, closely here, you can see that there are these little loop areas and there are these little grooves, and that's what allows it to bind to specific proteins on the outside of cells and let itself in. Okay, DJ1. Interesting protein for a couple of reasons. Number one, it was discovered back in 2003, and mutations in the gene that code for this protein are linked with autosomal recessive early onset familial Parkinson's disease, which means it's a recessive form of Parkinson's that if a person has both recessive traits, they get an early onset of this disease at a younger age. They do not understand, and they still don't know, even from 2003 to 2020, they still do not know the exact biochemical function of this protein. They've done lots of structural and sequence comparisons. They've looked at other ones to figure out what might be going on. They've identified the area that I highlighted in cyan. This is the putative active site. And what actually happens is two of these proteins come together and they form a dimer. Okay, and that dimer, made up of two of these proteins, acts to help reduce oxidative stress. Now, what does oxidative stress mean? Well, in the mitochondria, which is where this protein is found, um, there's a lot of oxygen in the mitochondria. And what happens is you get a lot of free radicals building up, and this helps to break down those free radicals, because those free radicals, if left to float around in the cell, can cause damage. They cause damage to not only the DNA, but they cause damage to cellular components like enzymes, membranes, and organelles. It's also involved in what's called androgen receptor regulation. And so this helps certain hormones like testosterone bind to the cell membrane and then pass through it. And without that androgen receptor, we have malfunctions in the cells. But this is a really interesting protein because it still doesn't have a known function. And that's one of the really neat things about biology. As much as we know about biology, and you know now with COVID-19, there are things we still don't know. We still don't fully understand the mechanisms behind how certain proteins work. And this is one of those examples, as is COVID-19. Okay, myoglobin. This is a huge protein to talk about. This was the first protein ever to be crystallized. Okay, and you'll see that when you start looking at the myoglobin assignment this week. So this is a close-up of what's called the heme group. The heme group is what carries oxygen. Okay, there's an iron atom right here in the middle that has a very high affinity for oxygen. And that heme group gets tucked right in here inside of this molecule. Now, if you look at this, this looks like almost looks like a crane. You know how you see a crane with all the extensions to it? This is something I thought was really neat. These are all the hydrogen bonds that occur in the alpha helices within this protein. Okay, so it's a really stable protein. It's got lots of hydrogen bonding in there, and I thought it was really neat just to see how this thing is set up. The other thing that's really interesting about this protein is the whole thing is made up of almost entirely alpha helix, um, which is kind of neat. And so what I've done here is given you just one piece of this, um, which is the active site here, and I use cyan. Again, it's a pretty bright, obnoxious color, so that you can see that this heme group here interacts with these side chains. These are the R groups. These are the side chains of the amino acids, and that's what holds it in place. Any mutation in here is going to cause a change in the structure, and it may not carry oxygen well enough. Okay. Um, Now, myoglobin is similar to other types of globins. There's an alpha and a beta chain for hemoglobin, and hemoglobin itself is made up of 4 chains. It's a gigantic molecule, and it contains 4 heme groups, and that's what carries the oxygen in our blood. Okay, so that's it for the myoglobin. That's it for the proteins that we've looked at so far. I just wanted to give you guys a brief recap of what it was that you were looking at, because we kind of got into it pretty quickly and we started looking at structures and never really got a chance to highlight some of the functions of what these things do. Okay, so I hope you found that helpful. Again, this is what your myoglobin is going to look like when you model it. Okay, and there's a nice quick little video on that and some background information. Okay, so good luck with everything this week. Reach out if you have any questions. I'm always around, you know that. And I'll see you soon. Okay, take care, guys. Bye.